candidate. This is Benedicte Bachelot. She had her PhD in Columbia, Columbia, not the country, sorry, Columbia University in 2015. Then she did her first postdoc at Duke from 2015 through 17. And now she's a postdoc at Rice. So help me welcome Benedicte.
Thank you very much for having me, and I'm sorry I'm very late again, but I'm very excited to be here finally and getting to meet all of you at Discover Campus. So I'm going to talk about my research. And specifically I'm going to kind of try to illustrate how I conduct research and how I'm helping my lab with conduct research. So in particular, I combine a lot of fieldwork and theory to address the question of plant ecology. So I will try to highlight some of that work. And then more recently I started doing more some experimental work because that's really the only way to really get the mechanism behind some of the processes that are plant ecology. So what I'm hoping is that I'm kind of a mixture of those three approaches, and that's really what I want my future lab to be doing, combining theory, experiment, and fieldwork to address major function in ecology. So we all know, since we're ecologists, that the world is changing dramatically. That's talking about deforestation, dramatic shift in the planet, the spread of disease, the rise of agriculture, and all of those changes have a very strong impact on our planet community. So one of the main goals of my lab, my research, is to understand how climate change and disturbance, so more generally, is altering species interaction that shape planet communities. But before you can answer that question, one of the things you need to understand is, well, what are the species interaction that shape planet communities regardless of climate change and disturbance? So today I'm kind of going to talk about those two questions entirely. So I'm very interested in plant communities that are extremely diverse. So because of that, I became a tropical plant community ecologist. And all those tropical rainforests where I did some work, you can get more than 100 species coexisting in one single hectare of forest. So an incredible diversity of species that more or less use the same resources. So that leads to the question, well, how can so many tree species coexist in one single hectare of forest? So not only those forests are extremely diverse, which is interesting for my question, but they're also extremely important. Right? They stabilize our climate, they maintain the water cycle, very good source of food and medicine, and they're very fun to explore. You might be tied in some sitting and then discover capuchin monkey, start like throwing some branches at you, for example. So let's say you get a chance to go in those forests and you start recording which species, tree species you encounter are what relative abundant, and you plot their relative abundance from the large scale, and you organize them from the most common to the least common. What you're going to see is a very long tail of rare species and a few very common species. So you need two things to happen to get coexistence. You need to prevent competitive exclusion, and you need to rescue all of those rare species that are extremely rare. So we know that negative density dependent feedback can do those two things, right? You can prevent competitive exclusion and you can rescue the rare species. So let's say you're very abundant. That means you're going to have a lot of negative feedback, so you can't really increase in size much more. But if you're a rare species, you have very low density, so you don't experience a lot of negative density dependent feedback, so you can increase much more rapidly in size. So you kind of escape this negative effects. So for the rest of the top, common species are always going to be in yellow, and rare species are going to be in blue. So in tropical ecology, plant tropical ecology, the Jensen canal hypothesis is one of those hypotheses that explain diversity, that is the foundation of this hypothesis is negative density dependent feedback. And the idea is pretty simple. You have a parent tree, and that's the seed input, so you get a lot of seed near the parent tree and not so much as you go away from the parent tree. And there's all those enemies, so pathogen, herbivores, that are thought to be to some extent, they have host preference, so they're really like this growing species. So you're going to get a lot of enemies near the parent tree and not so much away from the parent tree. So if you're one sitting, you're probably a reproducing is going to go increase as you go away from this negative pressure. And then the probability that one of the seedlings becomes the next tree of that species is going to peak away from the parent tree at low specific seedling density, right? And then you need ecological space for another species that is not sensitive to those natural mutual group. So you can see this process is going to promote coexistence at a local scale. And this is going to be one little symbol for local scale. So I'm talking about the few meters. And that kind of brings me to talk about scale for a minute. So when you think about coexistence, you already care about coexistence at the community scale, right? Not necessarily just the local scale. And it's very important to think about all those processes acting at both the local and the community scales. And there's many reasons, right? But one of the reasons that's important to think about is that the abundance of the species might be very different depending on the scale at which you talk, right? So at the local scale here, in that little seeding plot, so I'm talking about 1 by 1 meter, the green species is very abundant, and that's because I happen to be next to a very big green tree. But if I zoom out and look at my entire community, well, it turns out the yellow species is more important, right? So you already want to think about all those scales when you think about community ecology. And so throughout the talk, I'm going to bounce between those two scales, and I'm going to use two measures of the abundance of the species. At the local scale, I'm going to use coexistence density, so that's how many seedlings of a given species I have. And then at the community scale, I'm going to look at sum of that tree area. So that's now summing the area of all those trees. Okay, so the idea of the Jensen hypothesis is very attractive, right? Because you can easily measure conspecific seedling density, that's just counting seedlings, and then you can easily look at the seedling dynamics and how it changes with that conspecific density. But one of the main issues with that hypothesis to me is that it's a giant black box, right? What happened with conspecific seedling density? But there are a lot of things happening. You had change in competition, you had change in natural mean pressure, so that's one of the foundation of the hypothesis. And more importantly, and that people are tended to ignore, is that you also have change in mutualism. And that's extremely important when you think about all the tree species, especially the tropical tree species. More than 90% of them do associate with a very important type of mutualist, the Arbuscular mycorrhizal fungi. So those fungi are obligate mutualists, they cannot make their own carbon, so they go inside of the cell of the roots of the tree. They develop those little tree structure where they're going to exchange resources with the plant. So the plant is going to provide carbon to that fungi, and that's the only way the fungi can get carbon. In exchange of this carbon, the fungi is going to give a lot of micronutrients to the tree, in particular phosphorus, but also some water. So it's this like nice mutualism happening. And then last but not least, we've had more and more evidence showing that those fungi, those mutualist fungi, also change the defense pathway of the tree in a way that kind of like make them better defended against naturally. Okay, so we know most tree species are involved in those interactions. So if we go back to the Jensen hypothesis, mutualism was nowhere in the picture. And if you start thinking about, well, what happened if I start thinking about mutualism? Well, you can really change the expectation of the hypothesis. In particular, this is where there's a lot of faults, right? So there's a very strong potential at high conspecific seed density near the parent tree for those mutualistic association. And since the good association is going to boost the probability of maturing, which could mean that the tree is going to be able to recruit much more closer to the parent tree than it could do in the absence of mutualism. So you basically have this very strong potential for mutualism to totally counteract the Jensen hypothesis. And what that means to me is that we need to move away from just focusing on one type of interaction, and we really need to encompass all the interactions that exist at the same time. So that's what I did first theoretically, and I don't have time to go through the model with you, but I'm going to briefly summarize it. So if you think about your tree species, your plant species, they involve in competition, and then they're also being created upon by natural means. And then more importantly, they do customer resource type interaction with those mutualists. Right? So they give carbon away with their resource benefits, and the fungi can kind of change the way the natural may respond to the plants. Well, if you do that mathematically, you're going to get coexistence. But the very important question is, well, what are the conditions that are going to promote entry species from coexisting together? And it turns out the result of this mathematical question was that we need a very nice balance between the negative effect of the enemy and the positive effect of the mutualists. And if you have this nice balance, then you get in this sweet spot where you can have a lot of trees coexisting together. So that was great. Mathematically, we can prove that together they can lead to coexistence. But there are tons of assumptions in all the model, right? And I'm just going to list two that I'm going to talk about today. But there's like, a very long series of assumptions that came into building a mathematical model. And one of the assumptions that is very important is that those naturally do not respond nearly linearly to the abundance of tree species. And then another very big assumption in the model was for tractability and to be able to derive the math. I kind of ignore that there's different species of fungi. I just say there's one giant community of fungi that is beneficial. And I'll come back to those hypotheses. Okay, so the main question already, and the main way my lab is functioning, or the moment we find that interesting, is that I'm thinking about how mutualism influences very well known mechanism of coexistence. So I'm kind of trying to change the way we think about coexistence. And then I'm going to think about how all those interactions are changing in the face of climate change. Okay, so first of all, story I'm going to talk about illustrate some of my fieldwork, something that I would like to continue. And really it's like focusing on this first assumption, which is that the relationship between enemy and plants is not linear. And I'm going to ask, who are the above-ground naturally targeted, and especially how are they responding to the abundance of tree species? So if you think about all the ecological process that might be happening, you have a strong expectation for a non-linear relationship. So in that, I'm going to just make a few hypotheses. So enemy pressure is changing non-linearly as a function of community abundance, and this is because when you're rare, you might escape detection, right? You're just too rare for the enemy to find you. Or you might experience what we call heterospecific herd protection, which is you're the only one of your species that is surrounded by another species that might deter the enemies away from you. And then on the other side of the spectrum, if you're very common, a lot of things can be happening. There may be natural dissociation, right? They're not resource limited. You can have heap dropping among conspecific that kind of make you better and that's like responsive to attack. And if none of that works, you might have a third cosmic level that can control the population of matter. And if those are very strong effects, then you can even end up with some sort of handshake relationship. But in any case, we did not expect a linear relationship. So I had one question that's really a double question, which is how are the naturally responding to the local abundance of the tree species and how they are responding to the community scale abundance, so those two little scale I was talking about. So to do that, I'm going to focus on the seedlings, and that's because this is the life history, the stage of the life history of the tree that's extremely sensitive to biologic interaction. You get tons of data, and they're also a very good representation of how the future community of trees can look like. All the data come from Puerto Rico in the Luquillo forest dimension plots, which is a 15-acre long-term forest plot where all the trees have been monitored for many years. All of those symbols represent the location of those little seedling plots, and the seedling have been annually censused since 2007. All the environments as well known, so we have a very good long-term data set coming from Puerto Rico. So what did I do? I went to all those seedling plots, so roughly 237, and I'm not an entomologist or a pathologist or any of that. So I had to find a way to get a sense of what the enemy pressure was without being an expert in enemy. And one way I found was that you can borrow paleoecology technique, which is they have a lot of fossil of leaf, but they don't have the fossil of the enemy. So what they do is that they use the morphotypes, so the shape of the damage that they observe, as a proxy for species of enemies. So to do that, I went to all the seedlings in those plots, took pictures up to 5 leaves for each seedling, and then I processed all of those pictures to try to find how many unique morphotype of damage I found for each individual. So that's a lot of image processing manually. I moved away from that, so now I have a new script that's supposed to do that as well as I did. I'm working on it, but it's almost done. And then that gave me natural near richness, and then I tried to predict this richness using a Bayesian model as a function of range of variables. Okay, so who are the naturally targeted? So I'm going to show you a lot of those plots, the standardized coefficient plots, the variable in the model, the horizon on the y-axis. Then I'm going to show you the Crosshair distribution with the median and the credible interval. To be significant, you don't want the credible interval to overlap 0. If it's on the right, it means the covariance is associated with an increase in enemy richness. If it's on the left, it's associated with an decrease. So there's a range of variables. Seedling size is just a way for me to account for the age of the seedling, and as you grow older, you're going to accumulate more damage in your leaf, so you need to correct for that. Water flow and shade are just a way to correct for the fact that some pathogen might really like it when it's very moist, so it's kind of an environmental variable. But then the one I really care about is the local abundance of the tree species, so this conspecific seedling density, and I put a linear and a product of time to allow for more flexibility. Okay, so as expected, a bigger seedling, an older seedling, are way more naturally mean than a young one. That's because it's been around longer, so it's been attacked more. But then, very interestingly, the local abundance is significantly positive. So as you expect, if you're very common, naturally are going to defend you a lot. But the product of time is negative and significant. So what this means is that the common species species, so the ones that are very high cost-specific density at the local scale, they don't have as much natural mean as some of the more intermediate or rare tree species. Okay, and then the intercept of the model is mediated by the abundance of the tree species at the community scale. So this sounds about rare. And that allows me to answer the next question, which is, well, how are those same natural mean responding to the community scale? So this time, linear trend was not significant, but the quadratic trend was significant, and again, negative. And that was again driven by the common species. Somehow, it was costing less naturally than some of the intermediate and rare tree species. Okay, so what did we find? We found that there was some sort of Hampshire pattern, so there's not a linear relationship between enemy and plants. Abundance, and it seemed to be driven mostly by those common species, somehow not being targeted by natural mean as much as the intermediate or rare tree species. So we don't know why. That's something that I'd like my lab to kind of dig a little deeper. It's like, well, what if those common species somehow not as palatable or as interesting for the natural mean? There's a lot of things you can think about, but the first path to take out is just to look at what type of functional traits they have, or what characteristics of the leaf or maybe chemistry make them that they're not as interesting for natural mean. So that's something that I like my lab to do first. Okay, so we know that one of the assumptions of the model at least is not validated, so that was exciting. The next thing is that, well, the overall conclusion of the model is that I can get coexistence because of the action of enemy and mycorrhizal fungi. Well, can I get some evidence with real data that indeed you can get coexistence because of those two different type of agents? So this is what I did next. I asked about the natural mean and the mycorrhizal fungi interacting dynamic in a way that may promote or not tree species coexistence. So we do have natural mean data, I just talked about. We have stealing data because those are all long-term dynamic plots, so there's a suite of volunteers every year that come and get data for us. What I don't have is formal data. So for that, I went to all those seeding plots and I collected the soil sample at the corner of those seeding plots. So the seeding plots, they're very small, they're like 1 by 1 meter square, but they're long-term dynamic plots, right? So I'm not allowed to do any arms inside of the plots. So one way to still get at what are the fungi present in those plots is to just connect at the four corner and kind of homogeneous space and kind of assume they're a good representation of the fungal community. So I did that, and then I extracted the DNA from those seeding plots, and then we sequenced it using Illumina platform with a kind of broad fungal primer. Because I was not just interested in fungal, fungal, but also all the fungi present in the system. But today I'm just going to talk about the Arbuscular mycorrhizal. So they're a very nice group of fungi, right? Not only the mutualists, but more like practical, or maybe they're monophyletic group. So once you sequence everything and you get your giant composition matrix, it's very easy to pull up which one are mycorrhizal fungi versus non-mycorrhizal fungi. So there's the third more important group, not too surprisingly. But then you kind of have this like giant matrix of like, species, implication, different number of traits for them, and you kind of have to like summarize that information in a way that I can use in the model. So I decided to look at Arbuscular mycorrhizal fungal diversity. And what I'm showing you here is just a map, the same map, the scripting I talked about all the seeding plots. And I'm just kind of showing you, especially that there's a very wide, big range in the diversity of Arbuscular mycorrhizal fungi. So you have some plots like this one that only had one Arbuscular mycorrhizal fungi species, but then some other one like this one that has up to 12 different species. So you get a nice spatial variation in that, so that allows me to ask, well, how does that spatial variation influence seeding dynamics? Some of the variation is explained by that two colors that I'm showing you that represent the long-term kind of legacy, lineage legacy. I can answer some questions about that, but I won't talk too much about that. Okay. So we have two questions. What are the local effects of enemy pressure and Arbuscular mycorrhizal fungal diversity on seeding mortality, and how are these effects changing at the community scale? So again, I'm playing with this. So we have a few assumptions. Obviously, if you put a lot of damage to the ceiling, it's going to die, right? And then Arbuscular mycorrhizal fungal diversity, as it increases, it should decrease mortality of the ceiling. And that's because if you think about selection and complementarity, right? As you increase the diversity, the likelihood of having one very good mutualist might increase. Or if the plant is associated with several of them, you might have some nice complementarity effect happening. And then on the community scale, we were interested in, we thought that the effect of mycorrhizal fungal diversity would be much more pronounced for the rare species than the common species. And this is because if you think about rare species, they are very low abundance, that kind of like not very well clustered. So they don't have enough biomass to influence the below ground microbiome, right? So they may be very dependent on whatever microbiome is there. So in their case, maybe it's good to be very diverse. But the common species, they're usually pretty clustered, they have very high biomass, so they might be able to actually have strong fingerprints on the soil microbiome, and they may not care at all about diversity as long as they want specialized Arbuscular mycorrhizal. And that's kind of like, there's not a lot of data on this, but that's something that would be like fairly simple to test, and then like an enterprise could do that for us in your cases, for example. But that was kind of like the hypothesis to start with. Okay, so another standardized plot. This time, if the variable is significant on the right, that means the ceiling risk of mortality are increasing. If it's on the left, it means ceiling mortality risk are decreasing. We had several variables. Shared tolerance is just the intercept, and that's just allowing to take into account different life history strategy. Ceiling size is just to account for the fact that once you're big enough, you might not die anymore. But then the variable that we really care about are foliar damage, which is a measure of enemy pressure. Then constant ceiling density, that allows me to take into account those other black box elements that I didn't think about, so for example, competition, or below ground enemy pressure that I do not have that around. And then Arbuscular mycorrhizal fungal diversity. Alright, so what did we find? Not surprisingly, a big and tall ceiling has a low risk of mortality, but foliar damage and conspecific ceiling density both are significant and they increase the risk of mortality. So that makes sense. This is the Johnson banana hypothesis, and this is one kind of aspect of this black box, the fact that above ground enemy pressure participated in it. But then, very interestingly, Arbuscular mycorrhizal fungal diversity was also significant and negative, right? So if you're sitting in a plot that's very diverse from the mutualist point of view, you're less likely of dying. And what was very interesting is that the effect size is fairly similar to the conspecific of the foliar damage, but it's not bigger, right? So it's a little smaller. And that's kind of going back to the math, is that you want kind of a sweet little balance. You don't want one to be much stronger, right? If you have very strong mutualist effects, you kind of lose the negative density kind of feedback that you need to make coexistence. And if it's not strong enough, you might not get coexistence. So it seems that at the local scale, they kind of counteract each other, but not fully. Okay, what about the community scale? So for that, one way we decided to ask the question is to think about the data set and kind of split it into different levels. So data set depending on the abundance of the species, of the tree species, at the community scale. So either you're a common species, intermediate abundance, or rare tree species. And then we rerun the same Benedicte model. So I'm going to show you the three variables of interest, but this time three little cluster distribution for each of those three variables. Okay, so both foliar damage and conspecific ceiling density were only significant and positive for the common tree species, and then Arbuscular mycorrhizal fungal diversity was only significant for the rare tree species. So what that means is that at the community scale, they seem to be working together to promote coexistence. And what I mean by that is that the Arbuscular mycorrhizal fungi diversity was very important to rescue the rare species only, and then the enemies and conspecific density was very important to prevent superdominance. So you had this nice kind of like dual effect of those two agents that seem to be working together in a way that's consistent with coexistence. So that was great. But one of the obvious things with this, this observational study was that Arbuscular mycorrhizal fungi diversity is what really matters. Not just how much biomass of mutualist fungi I have, but really the diversity, the fact that there's different species that may be very important. And if you remember, one of the assumptions of the mathematical model that I didn't have time to present is that there was no species did not matter for mycorrhizal fungi. So that was a big issue in the mathematical framework that I would like, one of my students to jump in and kind of modify the model. And what that means is that the dimension of the model are going to explode. So it's going to be much more complicated mathematically, but I think it's worth investigating. So what that also told me was that the diversity of mutualists is very important. So then, how do you maintain the diversity of mutualists? And that's kind of a theoretical question that's not new. A lot of questions have asked this question. A lot of people have asked this question. And it doesn't, if you think about mutualism, it doesn't really make sense that you should have a diversity of them. And this is because, well, over time, the best mutualists should be selected, right? You should end up with only one super Arbuscular mycorrhizal fungi that's the best at giving you phosphorus, and that doesn't cost too much. But that doesn't happen in any mutualist system, that never happens. And then the other thing that is obvious of mutualism systems is that they're very sensitive to shears. So because of that, a lot of theoretical work, previous theoretical work has focused on, well, how do you stabilize those mutualistic systems? So that there's been a lot of work on sections, and then more recently, I think there's more and more work on the network structure of mutualistic interaction. But what hasn't been done yet is really thinking about those interactions between two partners as being very fluid. Right? It's not like a permanent interaction. Like I'm not going to have one partner and only going to interact with this one, but rather there's this idea that there's an ongoing partner choice. Maybe today I want to interact with you, but tomorrow I want to interact with someone else. So if you think about this ongoing partner choice, can this kind of very dynamic interaction promote coexistence in itself? So to ask this question, we had to think about different mycorrhizal fungi. And the mycorrhizal fungi are kind of different than the plant species because you can't really cultivate them. There's maybe 1% of all the existing fungi species that you can cultivate, but most of them you just don't know how they look like. But more and more there's been some work showing that it seems that they all have different functional traits, and in particular you can have like two big categories of fungi. You have what I call the cheap one, which don't have a lot of hyphae, so they're not very good at giving you benefits, but they don't cost a lot of carbon. And then you have the expensive one that invests a lot of carbon into hyphae, so they're very good at giving you a lot of resources, but they cost a lot, right? Because all that hyphae growth needs to come from the carbon you provide. So let's say we have a plant species that can associate either with an expensive and a cheap fungi to obtain nutrients. It can do it on its own, but it's not very good at getting nutrients on its own. So that's kind of thinking about tropical trees not being very good at getting phosphorus alone. They really need those associations with the Arbuscular mycorrhizal. Then the question of ongoing partnerships is really, what is this strategy of this carbon allocation? So how much carbon should the plant give to one or the other over time in a way to kind of maximize its own fitness? So really I'm trying to solve this function A of T that represents the carbon allocation. And what I want to know is kind of ongoing partnerships alone promote coexistence. And I really emphasize this idea that it's alone because there's some previous work that tried to address this question that had very complicated, beautiful model to represent nature. But in their model, they already included some things that would promote coexistence. Right? So they had maybe niche partitioning kind of hidden in their model. So here we had a model that's kind of deprived of all those mechanisms that we know can already lead to coexistence, and we ask, can ongoing partnerships alone do it? So you have to solve this function, which is complicated, but for us, optimal control theory exists, and that's something that a lot of economy people use, and a lot of people in fishery and wildlife uses. And really the way it works is you try to find let's say, harvest strategy that would maximize your profit over, let's say, 10 years if you're allowed to harvest something for 10 years. So applied to our question, what we want to know is what is the carbon allocation strategy that's going to maximize plant fitness over a specific period of time. So you're trying to defend this allocation strategy that maximizes this integral. And so we need to define the time. And in this case, we want a very long period of time to kind of capture long-term change in dynamics of plant and global balance. So I'm not going to show you the math, but what's really important from this analysis is that there's this context dependency of how the benefits and the cost of associating with the fungi change with nutrient availability. And in particular, with the model, plant benefits increase in the function of nutrient availability, a little more rapidly with the expansive fungi than with the cheap fungi. And then on the other end, nutrient benefits are kind of a decreasing function of nutrient availability. And where things become very interesting is where those benefits kind of switch from being either a net benefit to net cost or vice versa. And those we call them pivot points, because at those boundaries there's going to be a dramatic change in what the carbon allocation strategy should be for the plant to maximize its fitness. In particular, there's three very important areas. In the middle you have pure mutualism, right? Everybody's happy. At low nutrients really you have kind of parasitism, where the plant kind of suffers from the association. And then at high susceptibility, you have kind of the opposite, where the plants kind of pursue the fungi in a way. And depending on where you are in this framework, or like what the model is taking you for the dynamic, strategy is going to respond to maximize the fit, right? So if you're in the area, you don't want to associate with the extension fungi because that will really hurt you. Right? Okay, so yes, ongoing partnerships can indeed promote coexistence, and we did a lot of numerical solutions to investigate this. I'm just going to show you one example that I thought was very interesting. So there's three graphs. The first one kind of show you this optimal carbon allocation strategy. If it's 1, all the carbon is going to the expensive fungi. If it's 0, it's all going to the cheap one. And in the middle is the fungal dynamic, and then plant and nutrient dynamic. So in that scenario, the plants have allocated a lot to the expensive fungi, and that's because you're in the range where the expensive fungi is a very good mutualist. But then you reach some elements where you kind of switch from being a mutualist to a parasite. So in that case, the plant change and start allocating more and more to the cheap fungi as a way to kind of buffer against the mutualistic parasitic effect of the expensive fungi. So you kind of like all of a sudden decide to rescue the cheap fungi to kind of buffer against this negative effect of the expensive fungi. So that was very exciting, theoretically, because that's the first time that we were able to show that ongoing partnerships can allow for coexistence. But another thing that was exciting about this model is that you can really use it to infer some prediction along succession. But think about how nutrient stability is changing along succession and use that to make prediction about the relationship between plant and mycorrhizal fungi. And in particular, we thought early in succession, or when nutrient stability is very low, we would expect the fungi to be more like the parasites, and the association to go towards the cheap plant. And then later, when you have much more nutrient stability, the fungi might be more like resources for the plant, and this time you might find more association with the expensive fungi. And we did some work to actually test this idea, and we did find some evidence that in early succession of species, association with mycorrhizal fungi is actually more on the parasitic side than the beneficial side, and that's something that's also been shown in grassland. So there's some evidence that can validate the prediction of the model. Okay, so that's great. But what happens if you disturb the system? Right? We live in a world where disturbance happens on a regular basis. If you think about the forest as in Puerto Rico, every 10 years or so, a massive hurricane come and kind of like reduce the plant biomass. You also have many disturbances that influence nutrient availability. So what happens if you kind of put those disturbances on top of the model? So I'm just going to show you a couple of equations. So this is the plant dynamic. So there's some nutrients benefit from associating with the fungi F. There's the carbon cost from associating with the fungi F. There's some nutrients uptake that the plant can do alone, some competition, and then the disturbance is included with some sort of periodic function in the dynamic of the plant. And this is to kind of represent either the periodic hurricane disturbance or periodic logging activity that you might do in your forest and so on. For the nutrients disturbance, this time we decided to disturb the nutrients by adding some sort of like periodic nutrients inputs. So that could either represent periodic like you can deposit human from amendment in your forest, sort of like fertilization effect. Or in my forest, what's interesting is that we have those periodic Saharan dust that bring a lot of phosphorus into the tropical forest. Any kind of periodic disturbance that would do that. And then there's like a bunch of things that happen in the nutrients dynamic that kind of represent the loss at different levels. So you got the loss by leaching, or some uptake from the fungi, or the uptake from the plant. So this is our new model. It's kind of very similar, just called the little addition of disturbance. And we repeated the same type of analysis. And what we found was that you can start with the base scenario without disturbance, where the cheap fungi was excluded, for example. You add disturbance in the model, and all of a sudden, the cheap fungi is not excluded from. The plant is actually constantly changing its allocation to the cheap and the expensive fungi to kind of respond to those disturbances. And really what's happening here is something similar to non-relative linearity, where with the disturbance, the best mutualist is constantly changing and the plant is like dynamically responding to those changes. So that was exciting. But even more interesting was that in some cases, we started with a very nice system where without disturbance you have very nice coexistence of all the mutualist fungi. In this case, it's because the cheap and the expensive fungi kind of reach a point where they're basically indistinguishable for the plants. They're exactly the same mutualists. So the plant associates 50-50 from this carbon. And you disturb that, and all of a sudden the plant excludes the expensive fungi and only keep the cheap fungi. And And this is because if you look at where you are in the context dependency, the disturbance led the expensive fungi to kind of flip and become a parasite, so the plant did not want that fungi anymore. So again, it's going back to this little framework that I showed you where, depending on what does the disturbance to the nutrient availability, for example, if you stay here, not much is going to happen, but if the disturbance kind of push you on the other side of the pivot points, that's when you're going to see dramatic change in the coexistence of how the fungi. Okay, so that's kind of exciting. It's a little harder to test in nature, but that's something I would like my lab to do. And one first step is, before jumping into directly disturbance, I want to see what happened along succession, where you have more like very slow changes in nutrient availability. And like I was saying, one of the hypotheses that you get, you kind of go from parasitism to not parasitism. And that's something that we tested with some like fancy statistics, so it's kind of like I call it a conditional treatment. But we basically manipulated the data that I showed you earlier to see what would happen if there was no mycorrhizal fungi in that forest. It's obviously something I did not do, but you can statistically play with the data to do this kind of fake treatment. And what we found, I don't have time to do all of this, but the most important is this part where the early successional plant species really do much better in terms of abundance when you remove artificially the mycorrhizal fungi. So this is where, this is just playing with the data, and one thing I really want to do moving forward is actually do some real experiments where you can really start testing those ideas and kind of seeing how the plant dynamic interaction change or the succession, and then start asking about disturbance related to that. Okay, so we know theoretically what may happen with disturbance, but what about with real data? So in that, a last little story I want to talk about is about an experiment where we're actually looking at how change alter climate, so change in particular temperature, but we also happen to have a drop happening, so both drop and temperature influence living dynamics. And in particular, all those treatments are really changing the interaction that hopefully we find for coexistence. So the first thing is, if you think climate is going to matter, that's because it might influence the plant, obviously. But it could also have very strong influence on the enemies and the mycorrhizal fungi, so on all the partner hormone interactions. If you start trying to think about how those effects might be, you can pretty soon get crazy because they could go in every direction. And I'm just going to summarize roughly the main thing you might think about, which is warming might have kind of a fertilization effect on the plant, so there's a lot of nitrogen. There's also kind of like a fertilization effect on the enemy, so they might grow much more rapidly at higher temperature. But then the drought might have kind of a negative effect on the enemy. If you think about pathogen needed moisture to disperse, it could also have some inherent positive effects. So overall we think okay, maybe warming has a good effect on the enemy, drought has a negative effect. And then on mycorrhizal fungi, kind of similar, warming might increase growth rate, either directly by the effect of the fungi, metabolic activity, or indirectly by increasing the carbon available for the tree to give to the plant. And then droughts, we think, could have potentially a good effect on the hypothesis, and that's because it's been shown. But more importantly, it might more likely have a negative effect on mycorrhizal fungi that kind of negatively impacting the tree. So we had all those different ways in which climate can change mycorrhizal fungi community and natural community on top of influencing the plants. So what that means is that it's really hard to make a prediction about how climate is going to really change all those species interaction. But we still decided to look at it. So we first asked more broadly, without looking into their interaction, how drought and climate change influence seedling demography, and whether or not changes in enemy community and mycorrhizal community were responsible for some of those demographic changes. So to address those questions, I'm using the first warming experiment ever in a tropical rainforest. So in Puerto Rico, it's called TRES, where we're bringing some very intense power into the tropical forest, to those little hexagonal plots, and we're actively warming the understory to +4 degrees Celsius. So we have 3 little warming plots where we're actively warming the understory, and then we have 3 control plots. And the warming plots are constantly adjusting the temperature to be exactly +4 degrees Celsius compared to the current condition in the control plots. And then since we set up this experiment, I've been surveying all the seedling in the understory of those plots. So we had several years of data already. We have when we first set up the experiment, it happened to be the worst drought that Ontario ever had. So we had this nice, like, drought effect. We don't have a pre-drought treatment, sadly, because we didn't know that would happen. So we had a drought, then we had a post-drought, and then we turn on the meter. So we started having warming treatment. So we have about 2,000 seedlings from 43 different species. And annually, since we set up the experiment, I've been recording not just the seedling demographic, but also enemy pressure using photograph, and then also look at the root architecture of seedling outside of the plots, because I don't want to disturb the plots yet, when they're alone. And then I looked at hypothesis model to look at how seedling growth and mortality is changing over time. And I'm just going to show you mortality. And so what we found was that in the control plots, you get kind of typical Jensen-Connell effect, where you get increased mortality at high conspecific seedling density. But then in the warming plots, you get the reverse. You get some sort of positive density dependence, where the seedling die much less at high conspecific density than when it's not. You get this kind of negative trend. And then in the drought, there was just no significant effect at all of conspecific seedling density. So all what this is telling us is that in the essence of change in planet, you get the typical negative density dependence, but when you have drought or warming, you actually get an increase in negative density dependence that even switch to positive density dependence in the case of the warming treatment, which is very exciting in a way, because it's like a very strong effect. But it's also kind of scary, right? Because if you remove negative density dependence, that might be very bad for the coexistence and for the future of our forest. So, well, what could that be? Now remember, when you look at the overall effect of conspecific seedling density, you have all those things that happen simultaneously. So it could be that you have just a decrease in the enemy pressure, you could have just much more stronger mutualists for some reason, you could have a little bit of everything, they could be challenging the plants, shrek themselves. So it's very hard to it's going to take all of that, but we try to look at some of the data to try to see if we could at least find a reason for what we saw. The first obvious one is that, okay, maybe there's just no more nationalities, they just don't do well in the droughts, they don't do well in the warming treatments. But that's not true. So in the warming treatment, in that case, there's no change in how much damage you experience, there's no change in the diversity of the enemy that are observed. In the drought, it seems that there's something during the drought there is much more foliar damage, so that should not result in the decrease in negative density dependence. But there's also less diverse community. So maybe naturally we are doing something, but it's not very clear from this kind of overall picture. So we look more specifically at different types of enemy. So you have, for example, the gall makers, the endophytes, the grazer, leaf miner, and pathogen. And there's a lot of things going on here, but the main thing I want you to focus on is that both in the drought comparison or the warming comparison, we have some evidence of a decrease in pathogen populations. And that's kind of interesting to me because we know that Jensen-Connell hypothesis is mostly driven by some pathogen, right? It's not driven by the herbivore. It's really the pathogen that matters. So the fact that you get both in the drought and the warming treatment less pathogen tells me that, okay, that could be why we get this kind of decrease in negative density. What about mycorrhizal fungi? So I was not allowed at the time to collect in the plots. So I collected seedling all around the plots. And I looked at the root architecture either near the control plots or near the warming plots. And what I found was some evidence for important change in the root architecture of the seedling, where the one near the warming plots had much more biomass invested in the fine roots. So you have a lot of very, very small roots near the warming plots than you have near the control plots. And that's kind of consistent with the idea that you might have a strong increase in allocation below ground to the roots, which could be because they're allocating more either to the fungi or to some other microbiome. So we really excited. That's kind of exciting to us, and we're trying to push the analysis further to actually look at, well, do we have evidence for a change in mycorrhizal community, and are there some change in the root colonization? Okay, so we found that there was a decrease in negative density dependence, even switch to positive density dependence. And it's not very clear from just the observational data if it's due to the enemies of the Arbuscular mycorrhizal fungi. Likely it's due to a little bit of everything. And that maybe the Arbuscular mycorrhizal community is changing dramatically. But we don't have that yet. So the next step is really to take this, put it in the greenhouse experiment, and actually start looking at what happens when you warm the forest what happened to the fungal community, the enemy community, and try to like disentangle all of this. So to do that, I haven't submitted yet, but I'm leading a proposal for the NSF where we're going to actually try to get someone to really look at what happened when you walk the forest. In our case, it's after the hurricane. So what happened to the forest recovery following the hurricane if you walk or not walk? And in that pool of money, we will have a big rigorous experiment to try to disentangle the effect of naturally, the effect of mycorrhizal fungi, the effect of just changing nutrient availability due to warming and so on. So we're very excited about that. Okay, so that kind of like finishes the different story I wanted to show you to illustrate the different type of work that I do and how it's analyzed to do. Now, however, I convince you that it's very important to look at all the species interaction that happens mutualistically on your plants. And then a good way to do that is really to combine theory and data. So kind of not only use one approach, but kind of use all the approaches together. And it's kind of the perfect cycle, right? Where you like have some data, which is personal theory, and then the theory changes everything, and you kind of like constantly revising the theory and collecting more data. Now, I don't have a lot of time, but I wanted to like briefly talk about another thing that kind of fits in that talk, but not as well, which is a proposal that was submitted to CNH, which is called for natural and human system. That's kind of investigating plant community in a changing world, but kind of different. And that gets really interesting in the interface between coffee plantation and natural tropical community forest, and in particular, in this idea that fungi, so the mutualist fungi, but also the pathogen, are constantly flowing in and out of coffee plantation, going to the tropical rainforest, but also from the tropical rainforest to the coffee plantation. And we're interested in kind of characterizing those movements of fungi, either due to wind or due to bird movements. And what we're going to do is like catch a bunch of birds, swap them to kind of see what fungal is bringing in and out of the plantation. And we have this nice kind of gradient of different type of management, right? Either you shaded coffee plantation or you're not, and either you're like in the forested landscape or you're not. And we're kind of interested in seeing how kind of the human landscape is going to influence the movement of the fungi in a way that's going to really change the way plant communities are responding to fungi in the forest, but also how your coffee plantation is going to respond. So that, I'm very excited about that because it's a more applied side of my work, and I'm just showing that because I want my lab to be more, sometimes more, a little bit more applied. And we all need coffee, so. Alright, so without further ado, I'll take any questions.
So are rare tree species generally more flexible in terms of how many different mycorrhizal they can form associations with?
Well, there's not been a lot of work on that. I'm from the data set. Again, my data set, I don't know exactly who you associate with, but the rare species I found is like much more diverse. It seems to me to me that maybe. And there's also like just the idea that if you're rare, there's no point in think about systemization. It's very risky to be a specialist if you're rare, right? So that might just be that you just can't be very specific in my association. Yes?
So I was interested in the idea you said about heterospecific bird protection. And I wondered, I don't know very much about plant insect interactions, but to what degree does that hinge on highly specialized natural enemies and how does that read out in actual natural communities?
Yeah, so I think at the very beginning when the Johnson Applicable proposed, people already thought natural enemy was very specific at the tropics, and I think more and more data coming in from, a lot of data coming from Panama that show that it's actually not true. They're not necessarily more specialized in the tropics than in the parts, but you have very strong host preference. And that seems to be true. Like the common species seems to have more like some stronger tie to like some kind of more specialization than the rare species.
For the sampling of the mycorrhizal fungi across the ceiling population, do you have to test for density dependence among the mycorrhizal species?
We did a little bit of that, and there's some. So the short answer is like there was some evidence for that, but it wasn't really strong. So it wasn't also like the way, I sample, it wasn't ideal to test this idea. But we looked into it. We looked at roughly the antifungal community and tried to explain why you get species, where you get them. And really, for everything that's not mycorrhizal fungi, it seems to be mostly driven by edaphic factor. And then the mycorrhizal fungi were kind of like, it was kind of hard to explain them. You just don't have a lot of pores. They seem to be either they don't care too much. But you would expect them to really be dependent on the plant community, but it wasn't that strong of a signal, which has been kind of fun about it. But the other fungi, they don't really care about the more like soil characteristic.
This is kind of a related question, but you showed the results of rare species that can become more diverse in regions. So can you decouple their diversity of fungi versus like abundance of spores?
Yeah, so we tried to do that. I didn't show you, but we did some additional model to try to decouple those. And it seemed to really be driven by the diversity effect and not an abundance effect. And that's why I was saying, the original theoretical model, it's kind of like a big simplification, and already species do matter. And that's something that I would change.
All right, answer all the questions, wonderful.